gosh, those are short.
Okay, so we're getting started on the nervous system in Module 7. Module 8 and 9 are also on the nervous system. So we're going to spend a lot of time on the nervous system.
They said they're coming, but they'll be a little late.
Okay, so what we're going to be looking at today is sort of the basics of the cellular level, the cellular anatomy, if you will, of the nervous system. Now, let's look at our timeline. Next week, we don't have class next week because we're on Thanksgiving break. The weekend after that, we will have a lab on Module 7. Now, there is no lab for Module 7. There's a few slides, but we'll push those slides to Module 8. And that's because for Module 7, we have a murder to solve. We're going to have our murder mystery. So when we get back together in 2 weeks, you'll have a quiz on Module 7, and then we'll have a murder in the room, okay? And we'll figure that out.
3 weeks between Thanksgiving and Christmas.
So yeah, so 3 weeks between Thanksgiving and Christmas. So our first week back, we have Module 7, lab and quiz. The next week is your midterm. You'll come in, you'll take your midterm, you'll leave when you're done. Okay? The week after that, our last week before Christmas, we're going to have a lecture on Module 8. So when you come back in January, the first thing you'll have is a quiz and lab. You'll dissect the brain in our first meeting back in January. Is that all good?
Whose brain?
It's a sheep brain. They don't let me have human brains. Yeah. So, all right, are we
Oh,
good?
they just said it's a she brain. Like
Sheep, lamb.
I was like,
Oh, they're little lamb.
Then
Yeah.
do we have like a study guide put together?
You know what? I feel like I saw something somewhere.
Yeah, it would be so helpful.
Yeah. So I will tell you, it's modules 1 through 7. Okay. Old quizzes are really good to study. I believe I wrote the midterm. I think so. And so therefore, it'd be when you have the same writer for your quizzes and your midterms, it kind of helps. You got the same kind of mood, if you will, style of questions. It is short answer and multiple choice. All right, I'll work on seeing if we have any study guide or something, okay? All right, here we go. Starting in your lab manual, we're going to be looking at the two main portions of the nervous system. We have the central nervous system, also known as the CNS, and then we have something called the PNS, or the peripheral nervous system. So CNS is the brain and the spinal cord. Okay, the brain and the spinal cord. This is what we'll be covering in Module 8. All right, in Module 8. You can see in our picture right here, we have the brain, we have the spinal cord going down like this. There are those nerves going off to the side, but that is not part of the CNS. That's part of the PNS. So CNS, brain and spinal cord. The peripheral means to the sides, right? Peripheral vision, right? Is going to be the nerves and the ganglia associated with the nerves. You just write down ganglia for now. We're going to study the peripheral nervous system, which oddly is the more complex part of this deal. You would think the brain would be the most complex part, but actually the PNS, because it has so many divisions. That's Module 9. Okay, so in Module 9 we'll be back to the PNS, looking at nerves and ganglia. Although we will look at the structure of nerves today. We'll see what those look like today. Your first blank there about spinal nerves. Is that what the blank says? Something like that.
Two types of nerves and peripheral nerves.
Okay, in those two boxes right there. The first box you're going to write down spinal nerves. There are 31 pair. 31 pairs, so 62. 31 on each side. Those go from the spinal cord outward to all your sensory organs, namely your skin, this kind of thing, right? So touch and all the kind of senses we'll be looking at. In the second box, we're going to say cranial nerves. There are 12 pair of those. Those originate from the brain and come straight down and affect something down in the body, straight from the brain. You can see those up in the picture here, straight from the brain, the ones coming straight out, right there. And then the yellow ones coming from the side, those are all of the spinal nerves. Okay, those two boxes good? Now something I need you to remember in my effort to try to keep things understandable, I use this word action, I use the word signal for action potential. Okay, so remember we're talking about a signal traveling down in there. Remember we talked about signals coming to your muscle and moving your muscle? Comes from a neuron, right? So we'll be looking at neurons, and when I talk about neurons sending a signal down them, it's called an action potential. That's the official name. Sometimes I'll call it an AP for action potential. But I like to use the word signal because I feel like we understand that one better. The signal's coming through a wire, yes? So I'm going to be using that word. Just keep in mind that an action potential is a signal. All right, somewhere in your thing, in your notebook right there, maybe at the top, just write down that the functional unit of the nervous system is the neuron. A neuron's a cell. It's the cell of the nervous system. And our neurons are, I'm gonna have y'all drawing a picture of these here, or labeling a picture of these here in just a minute, because they're that important, okay? They look funny. You know, normally when we draw an animal cell, we start with a circle, we put our nucleus in there, our little mitochondria, our endoplasmic reticulum, all that kind of stuff. Well, this is not a circle shape. This is kind of a star shape with a long tail on it. And that's the shape of the cell. It's pretty cool.
We drew that a few weeks ago.
Did we?
Yeah. Yeah, we drew that exact. The motor neuron.
The motor neuron? Yeah, right. So now we're going to be looking at it from the perspective of the origination of the signal rather than the accepting of the signal, which the muscles do, right? Remember, there's two types of excitable tissue, muscular tissue and nervous tissue, right? Okay. Just know that's the functional unit. So we have these two types. We have the word afferent and efferent. They're very similar to one another. We got to keep those straight. Okay, so afferent neurons carry signals, that's the AP, right, the action potential, from sensory organs to the CNS. And the efferent neurons carry signals from the CNS to the effector organs, whatever needs to be. The brain's telling your body to write with your pen right now, so your effector in this case would be the muscles in your hand, the muscles in your eyes, your sensory organ of your ear listening to me. So afferent goes from sensory, here it looks like on our picture right here, we're looking at skin. It carries it to, this is the spinal cord right here, the spinal cord will carry that to the brain on an afferent nerve. Then, for instance, if on my skin I feel something, burning me, then my efferent nerve will send a message to my muscle to pull my hand away from whatever's burning me. You get it? Two different pathways. So the afferent says hot, the efferent says move. Get it? All right. We're gonna revisit this afferent efferent here in just a minute, and so it's important to keep those straight. All right. I have something for you. I have two handouts I'll be giving you today. This one's homemade. I made it myself. To try to make things understandable, it's going to be explaining this. The very top is cut off. It should say peripheral nervous system at the very top, but in order to fit everything on my copy machine, okay, listen, you need to keep this somewhere safe because this is going to be very important for us in Module 9 as well as this one. Okay, so let's take a look at it. On our picture on the slide up here on the PowerPoint, you can see that we're only looking at the divisions, well, I say P and S divisions. Our paper that I handed to you just now only has the peripheral nervous system on it. So notice right here we have central nervous system. All right, remember that's going to be Module 8. We're just going to ignore that for now. Okay, now on your thing right here, the only thing on the PowerPoint is the right side, right? Yeah, the right side of the thing I just gave you. Let's look at the left side of your sheet real quick. Notice it says afferent. Do you see that? That's the afferent division of the peripheral nervous system. Yikes. All right, notice at the very, very, very top it says sensory division. You see that up there? So afferent, I put a little memory thing on there saying a feeling. All right, so the afferent is all about from the organ to the brain, our sensory receptors. See it? We have our simple sensory receptor, which is the sense of touch, which is not simple at all. Bless you. And then our complex smell, balance, hearing, vision, taste. Do you see all those? That is all Module 9. Okay, Module 9, I think. I don't think it's 10. Yeah, I think it's module 9. Now, we're going to pay attention to the right side of the chart over here. Also for module 9 as well. Efferent. Efferent, I have on their email. It's like an email. Try to remember what efferent and afferent, okay? That's just a memory for me. I don't know if it helps you. This goes from the brain to the organ. The brain's going to send out an email. And we're going to have the two major divisions. We have the autonomic nervous system. This is called the ANS. It's involuntary. Okay? You might want to think of autonomic as automatic. Our heart is beating automatically, right? That's the autonomic nervous system right there. The other side is called the somatic motor nervous system, or the SMNS, right there. And that is our voluntary muscles. So that is taking care of skeletal muscles right there. Notice that our autonomic is divided into two more things. We have the sympathetic and the parasympathetic. That's going to be module 9. We don't have to worry about that right this minute, but you'll keep this when we get to module 9. Do you get it? So I don't know if this chart is helping you, but in your book, this was going to go somewhere. This was going to go, okay, because you see where it says two divisions, sympathetic and parasympathetic on your chart on page 40 of your book. Okay, so here's what I want you to do for this right now. On autonomic nervous system consists of, say, sympathetic division. Do you see that? I want you to write on there stress. The sympathetic division is responsible for times of stress. You see that on your chart. Sympathetic. Whole body, stress, and at the very bottom, you might not can see it, you might want to write it on the very bottom of your chart under sympathetic. I want you to write fight or flight.
Oh, right on this?
On this chart.
Sorry.
At the bottom of sympathetic, I want you to know the word stress. Notice S for sympathetic, S for stress. Fight or flight. Fight or flight. That's what you're putting here under your sympathetic division of your autonomic nervous system in your book. On page 40, you're writing stress under that one. On the next one, parasympathetic division, you're writing rest. Rest.
You already have rest written on it.
I know, but in your page 40 of your student book. I know. So all of this right here, they are answering these questions at the bottom of your book. In fact, in your book, on page 40, in that little box, when it says sketch summary of nervous system from PowerPoint, that's this. Right in that box, chart that Mrs. Casper gave me. Chart. Look at chart. Is this called a chart? Do you think this is called a chart?
Yeah.
Okay. So you don't have to write all this down in that box. Yay, I did it for you. Okay?
So we don't need to write anything in somatic, motor nervous, or automatic nervous?
Under somatic motor nervous, put SMNS, somatic motor nervous system, dash voluntary. And under autonomic nervous system, put ANS, involuntary. It's all here, I realize. But that should all point you back to the chart that I gave you, okay?
So the SMNS is voluntary.
Yes. The M in there, Donna, the M stands for motor.
Okay.
That means moving my muscles, means voluntary.
Okay.
Autonomic, the A starts for autonomic. Remember, we can think of it as automatic, but just don't put that in your quiz, please. Put autonomic, please, not automatic.
Oh.
Just put auto. So I'm okay with auto, A-U-T-O. All right, and so our afferent and efferent divisions of our peripheral nervous system are all on our chart right here. Afferent to the left, a feeling, all your senses. Efferent to the right, an email from your brain to your effector, okay? So you might want to keep this out. Although really, this is going to be much more useful for us in module 9. You'll want to have it out for that because it's going to go through all those little details, myelinated, all that business right there. Okay, so now you're going to turn your page to page 41. All right. And you're going to sketch a neuron of the peripheral nervous system, but we're going to skip that for a second. Am I on the right page? Okay. See in that box where it says sketch the 3 types of neuron structures? That's what's next. These are our 3 types of neuron structures. All right. you're gonna, in this box, you're gonna draw these things: bipolar, unipolar, and multipolar. And on the right here, it says neurons have different shapes depending on their function. All neurons have only one axon, however. There are approximately 100 billion neurons, and we lose about 7,000 a day. That's going to go under a blank called interesting facts. So sorry about the mix-up in the order. We're going to end up drawing one that looks like this multipolar one here in a little bit. Gabby, we're on page 41. And they're drawing these pictures in the box set that says 3 types of neuron structures, figure 7.4.
Sorry, we're late.
All
Sorry,
right.
Frank. No. I'm not ready for another one. Don't forget this is for you.
Thank you. Remind me, Gabby, did you show me a study guide for that?
Yes, ma'am.
I want to be wearing perfume because I walked in and it smelled really good.
Mm.
Smells
Okay, so on our neuron types that you're drawing right there, those 3 things, you want to make sure and put the label beneath them, okay? Bipolar, unipolar, and multipolar.
I'm not done yet.
I'm not
You're about
done
to
either.
change it.
I'm not going to change it.
Okay.
I'm just reminding you to write that there, but I want you also, those words on the right are going to go under the blank that says interesting facts, which you skip down one and the next one, I think. And then we'll go back to our neuron here in a minute.
The last one is
I know.
like the.
Wait, do we have to draw these?
Yes.
Yeah, and label them.
Right here.
Okay. Oh my gosh, this is
Not fun.
little thing.
Oh man. A lot of times really bad. I was doing so good. Ooh.
Are we still getting a boy in our class? Please say no.
Still what?
Are we still getting a boy in our class? Please say no.
I have not heard from them, so I'm guessing no, unless they want to start in the next semester or something.
Oh, great. I don't want a boy in our class. I like this class just the way it is.
Boys are gross.
I went to school
I know,
with him
right?
so many years ago. That was so weird if he was in my class.
Is he like
His
an older
brother.
I've done swimming with them.
You want everything in the bottom?
Just said interesting facts. That their shape is dependent on their function. There's only one axon. I think that's important. One axon. That's that long thing coming off of it. We'll get to that here in just a second. We have about 100 billion neurons, and we lose 7,000 a day.
Really?
So I'm a lot older than y'all, so I've lost a lot more brain cells than y'all have.
Oh, these are brain cells?
These are brain cells.
Ooh.
Yeah, brain and nerve cells, depending on where we are. But the neuron is the functional unit of the nervous system. Both the central nervous system and the peripheral nervous system Okay, so
Man, by the time you're like 100, you practically have like 2 brain cells.
actually we have so 100 billion is a huge number. That's a big number.
It said in our book that even if you live to be 100, at losing 7,000 a day, you've only lost 0.3%.
Exactly. So we're doing just fine. So don't. But there are things that can cause you to lose your brain cells faster, like sniffing glue. Don't sniff glue. It kills brain cells. And when brain cells die, they're
And
gone
Sharpies,
forever.
Don't Sharpies.
Anything that has that very aromatic, like
You're
the
already
gasoline,
sniffing
when you're putting
Sharpies.
gasoline in the car, you know it smells good, you know? If you smell a lot of that, can kill some brain cells.
that's
Certain drugs, smoking pot kills brain cells like crazy. People talk about how pot is not damaging. Oh my goodness gracious. That's why they all turn into potheads. They're dummies. Alright. Now, that doesn't mean that marijuana doesn't have beneficial things. I think it does. I think that CBD probably does, but not the part that makes you stupid. Part that makes you
Well, I
high.
feel like if you're doing it for a medical reason and not just doing it.
Yeah, you're probably not getting the
Pot for
stuff that
medical
makes you.
reasons.
There are people who have seizures and things like this, but now they're learning how to get that good part of it out and leave the stupid
Good.
part behind. Yeah. So the CBD, it's really big right now. CBD,
Wait, so this might
cannabinoid
be
oil.
a stupid question, but I'm gonna ask it. We have a friend who was just in a bike accident a few weeks ago, or like 12 days ago, like a little over 2 weeks ago, or whatever. Anyway, 1 week ago, and he's in the hospital, and he's been like completely unresponsive for 11 days. And yesterday, he just has really bad brain swelling and brain bleeding. And yesterday was the first day of him being responsive, but he's not like talking or anything yet. So he just is like making faces or whatever. So does that mean because he's unresponsive, he's losing brain cells?
Well, it depends on where the damage took place. So I'm sorry to hear that. Was it a bicycle or motorcycle?
Bicycle.
He got hit?
He hit a deer.
Oh no.
Yeah.
How do
He's
you
a
hit a
cyc-
deer?
He's a cyclist. And
Was
he's
he wearing a
going
helmet?
Really fast. We don't know, but we assume so because he's a cyclist.
So he's probably safe that way. Has anybody mentioned his EEG, his electroencephalogram? Nobody said the words flat EEG. Okay. Flat EEG would be bad.
I don't think
It needs to have some electrical activity happening in the
Yeah.
brain. If it goes, then that's bad. So it just depends, yeah, where the damage was done. If there's a bleed on there, they can drain that off. They're probably doing that, you know?
Yeah.
So when you say responsive in opening his eyes?
Yeah, like he opened his eyes yesterday, and then he can like he watched a video that his daughter made for him and made like a typical face that he always makes. And
That's
he's
good.
not,
That's a good
like,
sign.
talking.
Moving his hands, moving his feet.
Yeah.
Hey man,
Okay.
that's one good nap.
Yeah.
I will tell you
Yeah,
that
right, yeah.
neurons are the type of cells that the parenchymal cell, remember the parenchymal cells, the ones that do the work, right? That are permanent. So and that means if they're damaged, they never come back. Never, ever. So brain damage is permanent.
Did you say that gasoline can be one of the things that like
Yeah,
kills them?
they can kill, cause anything that's aromatic like that, and you smell it and you go, oh, and if you were in the room by yourself and it would make you high,
No.
that's killing brain cells.
Because I saw a video of a girl who was addicted to it and would take like shots of it and would keep it in like every
Wait,
room of her
shots
house.
of gasoline?
Because she like got addicted to the smell and so she would drink it.
That's poison.
Yeah.
Did it kill her?
I don't know. It was just a video of her. It was on like one of those My Strange Addiction things. and
Okay,
she was
so I'm telling you,
addicted
no, don't do that. Don't smoke pot.
to it.
Oh yeah.
Furthermore, don't smoke cigarettes. They wreck your lungs.
And they smell disgusting.
Okay.
Oh.
Next slide, y'all, goes up to the box above. The box above, but here's what I'm going to tell you. Don't draw that, because I'm going to give you this. Okay?
Oh wait, I have an old picture.
And furthermore, don't write anything just yet. Pencils down, pencils down. Okay, so remember neurons are the functional unit of the nervous system, and I promise you we're gonna write everything, we're gonna fill in every blank, I promise. But just don't do it yet. Okay? Trust me? Alright. So we're looking here at a neuron, and just to be technical, this is in the picture in front of you is a neuron from the peripheral nervous system. The neurons from the central nervous system look a little different, and I'll show you a picture in a minute. Okay. So this is a peripheral nervous system neuron, and I'm going to label the parts, but you're not going to label them yet. Okay. So the part right here that looks like a tree is called a dendrite. Don't label it, Gabby. Put your pencils down. All right. Dendrites. Dendrites mean having to do with a tree. It looks like a tree trunk or tree branches or something like that. Then we have the long purple thing underneath these yellow things is called the axon. Remember I just told you All of our neurons only have one axon. They have lots of dendrites, but only one long thing leaving called the axon. The body of the cell is called soma. The word soma means body. That's the body of the cell. What's in the body? The nucleus. What's in the nucleus?
DNA.
DNA. Thank you very much.
I've heard that clothing brand is called Soma. It's body.
What brand?
There's a clothing brand called
Oh
Soma.
yeah. They have really great underwear.
Yeah.
That's my favorite underwear. It means body. Yeah, it means body. At the end, we have the axon terminal that does branch out a little bit, and those are called the terminus or the bouton. Bouton means buttons in French. Then we have a cell inside here, that little purple part right there is a cell called a Schwann cell. We'll get to it, don't worry about it. And the Schwann cell is making something, and it's making this yellow stuff called the myelin sheath. Now, I reminded myself here to give you all the handout, which I have. The neurons are the functional unit of the nervous system. Their structure is unique and highly specialized. The dendrite brings the message in. The AP, the signal, comes in through the dendrite, and it carries through the axon and carries that away. The soma has many mitochondria and Golgi bodies. Mitochondria makes what? ATP, thank you. And the ATP is being carried out of the cell, and with the help of the Golgi body, which synthesizes, folds it in the right way, and sends it on down the axon. Okay. In other words, these cells need lots and lots of energy to do what they need to do. Lots of ATP to do what they need to do. All right. So this is what your thing looks like. Okay. Notice it's going the other way. I could have switched it, I guess, but here it is. And I'm going to put this up, and y'all are going to fill it in now. There. Can y'all read those words? Are they big enough?
Yeah.
Sorry.
I'm
Yeah.
like, no, it's fine, and then I actually.
Oh my gosh, I have dyslexia. This is not helping.
All the way over to the right, that one blank over there is soma. That's the body. And then our nucleus is our circle inside our soma. All of these things coming out right here, blah, blah, blah. Those are all of our dendrites, top line, dendrites.
wait. D-R. After D-R-I.
These are the dendrites, D-E-N-D-R-I-T-E-S. This was how the size they were on that page. I should go in with a box and make them bigger. I'll try to remember to do that.
Is that W-A-N-N on Schwann?
First one, S-C-H-W-A-N-N, Schwann. It's named after Dr. Schwann.
I was gonna say.
Dr. Schwann.
Just Ron Swanson.
The axon, I need to understand the axon is underneath these chunks here. So it's that thing running like that. It's the arrow is pointing to it right there.
Can you say what's underneath this Schwann?
This right here?
Yeah.
Okay, it's called a node. In between each one of these, the Schwann cell is making myelin sheath. Okay, myelin sheath, that's important. In between each one of them is called a node of Ranvier. So node, N-O-D-E, of, and a person's name, ready? R-A-N-V-I-E-R, Ranvier.
Where's the A?
Well, it's French. It's French. In French, you never ever pronounce the last letter
Is there
of a
an R
word.
at the end of
It is. It is.
Really?
Yeah. In French, you don't pronounce the last letter
Of
any
any word?
unless unless the next word has a vowel in it.
So, Abby Benoit, her name
I
after
don't
the
say
T.
Benoit.
Yeah.
I did.
I say
Ranvier.
When I first
It's
walked in the
R-A-N-V-I-E-R.
Benoit, and she's like,
Ranvier.
This is Benoit. And I was like, oh.
Whoa, okay.
But I still call it that as a joke.
S-H-E-A-T-H.
Sheath. S-H-E-A-T-H. Sheath. Myelin sheath.
Okay.
That's what the Schwann cell is making. Myelin sheath.
Cell / Benoit. I was like,
Benoit.
Benoit.
Oh, I would feel so bad if you said it like that.
You know,
No, I'm saying if that's how you said it. Like, my
That's-
last name's Benoit. Like,
have y'all ever heard of the city called Detroit?
huh?
You heard of the city called Detroit?
Yeah.
It should be pronounced Detroit. Detroit.
The funny thing is that she went from Benoit and then like a normal self and then to Pressoir. So like
I didn't know
Benoit
the new
self,
name.
Pressoir.
Pressoir, O-I-R?
Pressoir, yeah.
Oh,
Yeah.
Or
keeping it French.
P-R-E-S-O-I-R.
So wait,
Oh.
the node
Ranvier.
Ranvier makes the myelin sheath.
No. The Schwann cell makes the myelin sheath.
Oh, okay.
The Schwann cell makes the myelin sheath. The node of Ranvier is just a little gap between the. That node's going to become important to us pretty soon. All right, so listen. I gave you that sheet to fill out, yes?
Yeah.
Does that maybe give you the idea that that sheet is important to me?
Yes.
Yes.
Okay. Also
The
our
fact that you took
chart.
time to put it together, yeah.
All right, so hint, hint. Remember, in 2 weeks we'll have a quiz over that.
Oh, dang it.
So-
Wait, what did you say?
So, everybody have it filled in?
Okay, what is this?
Yeah.
That's our chart from earlier, talking about the divisions of the peripheral nervous
So
system.
do we want to put?
In
Gotta
this
be
box,
kidding
but
me.
we have a chart.
Yeah, in that box you want to write, say, see handout.
What?
Check
And
handout.
sketch a neuron in the peripheral.
In the box at the top of page 41, sketch a neuron, a peripheral nervous system, say,
Probably the worst
see
thing.
handout.
Amanda, write that.
So you didn't have to draw this whole thing. It'll take too long to draw. All right, the next thing is called an association neuron. An association neuron is simply a neuron that conducts signals from one neuron to the next in the CNS, in the central nervous system. In other words, it's neuron to neuron. We talked about neurons touching muscles earlier in our previous module, remember? And that axon and all that business, and that synapse and all that. Okay, an association neuron just means a neuron touching another neuron. It's essentially all of the little extension cords from my brain to the tip of my toe. I've got all these extension cords sending that signal all the way down to the toe. Although some axons on some neurons are really, really long. By the way, interneuron is another word some people use for that. It's used interchangeably, but I don't think your book mentioned interneuron. So just so you know, if you hear the word ever, someone says interneurons, they're talking association neuron. A neuron that touches another neuron. Sends a message down this neuron to this neuron, which sends it down to another neuron, ultimately to a muscle. It's going to cause the muscle to move. Okay? It's the reason why they're in there. Okay, so neuron to neuron is what an association neuron is. Yes? Okay. All right, there are two types of nervous tissue. We have neurons, which are the functional unit, the cell body and all that business. And then we have these weird little cells called neuroglia. Neuroglia. Okay, so I told you a second ago that the parenchymal cells of the nervous system are the neurons, and they're permanent, which means if they're damaged, then they do not get replaced. So neurons do not undergo any mitosis. Once you've got all your neurons, you're done. And then all you do for the rest of your life is lose them, 7,000 a day. Okay? There's no new ones. There's one exception to that, and that's in our nose. Our smelling neurons that allow us the sense of smell, those will regenerate. Those are the only ones. Okay, we'll get to that when we get to module 9.
Don't lose our sense of smell.
Correct. Yeah, we keep replacing
One of my
those.
friend's dad lost his sense of smell.
Permanently?
Yeah, because he got into an accident and
Okay, what that probably means is that the actual area that makes the cells was damaged.
Wait.
Or like if somebody can't breathe through their nose. I knew a guy in high school that had his nose broken so many times because he was a boxer or something that he really couldn't breathe through his nose.
Yeah.
Can't breathe through your nose, you can't get anything out
We
there.
had
So.
we had a friend who didn't, or maybe you were there, who was born without smell.
Interesting.
Does that mean she did not have?
It depends. It could be that those neurons are not there. It could be there was a disconnect between the neurons and the brain. That's a bummer.
Yeah, she's never smelled anything.
That's
like
very.
It's really sad.
Which
We
means
were like,
food.
oh my gosh, this perfume, because we were at a camp, and she's like, really?
Furthermore, she probably doesn't
Food
enjoy
tastes
food
so
as much.
different. Yeah.
She probably
Yeah,
doesn't
she
enjoy
doesn't like-
food.
I
You
thought
walk
that
into
would help with
Starbucks, even if you don't like
vegetables.
coffee, you walk into Starbucks, go, oh, it smells so good.
But I bet that would help with like vegetables and stuff that
Yeah.
have like
Like
a taste that you don't like she wouldn't taste as much.
You'd still probably taste the bitter, yeah,
Like-
of broccoli, yeah. Okay, all right, so remember I told you these don't regenerate, so therefore, have you ever heard of somebody who had brain cancer?
Yep.
Okay, brain cancer, any kind of cancer, is the over-mitosis of a particular cell. Well, it's not the neurons that are doing it, it's the neuroglia that are doing it. These are what turn into a tumor sometimes, the neuroglia, okay. All right, so our neuroglia are the helper cells of the nervous system. So here's our neuron. Here's our neuroglia. All right, they're the helpers. Those are ones that are doing the support work, right? The support work. So we're looking at the nervous system. The parenchymal cells would be the neurons, and the stromal cells would be the neuroglia, to take us way back to module 4, I think that was. All right, so also notice the neuroglial cells are small. They're small, and they have specific duties. We're going to go through each one of them here. I think you have a chart, yes?
Yeah. It's,
Are we ready for it?
The 5 types and
Okay.
then its functions.
We're going to be looking at the first 4 types. These are the ones that are found in the central nervous system, in the CNS. So the first one is called oligodendrocytes.
that's a fun one.
I always try to call them oglio, but it's oligo. O-L-I-G-O. Oligo. Dendrocytes, Dendrocytes. Oligodendrocytes. And
So
you can
then-
see a picture of it where? Right here. Okay, we're going to get to this in a second, but let me just tell you what this is. The oligodendrocyte is the CNS version of a Schwann cell. So that Schwann cell and that myelination, that's only in the PNS. In the CNS, it's called the oligodendrocyte. They're only in the CNS. They are the myelin around to insulate and bind CNS neurons, central nervous system neurons. Our next one is called the microglia. Microglia.
All these sound like a disease. I'm sorry.
Yeah. The microglia are responsible for fighting infection. They fight infection. They will literally destroy bacteria. There's very few brain infections. We don't get many. In fact, typically if somebody has a brain infection, it's actually an infection of the surrounding membranes of the brain called the meninges. You've heard of meningitis, right? That's not the brain exactly, it's the thing that holds the brain in place, which we'll get to in Module 8. The next group are called astrocytes. Astro means star, so they're star-shaped. They form something called the blood-brain barrier. All right, this is just a barrier to keep blood from touching the neurons because blood will destroy neurons. That's why if you have a brain bleed, the blood into your brain actually destroys brain cells.
Okay.
Yeah, it's bad news to have brain bleed. All right, so the blood-brain barrier protects it from these things. Now, there are some things that cross the blood-brain barrier. In fact, most medications that we use, if you have a headache, for instance, your neuroglia cells are aching. Your neurons don't have any feeling, but your neuroglia cells do. And so we got to get something to stop those from aching, and so we take a medication that crosses the blood-brain barrier and does it. So those are things that will cross it. When you sniff that glue, when you smoke that pot, guess what it's crossing over? The blood-brain barrier. It sneaks past. That's why it makes you high. Alcohol, it makes you high because it's going past that blood-brain barrier. Anything that passes through the blood-brain barrier can be a problem for your brain. That's why, for instance, you don't want to take ibuprofen every day of your life. You know, in fact, if you have a headache, try everything else first, your essential oils, you drink a big glass of water, peppermint oils are perfect, and then finally take your whatever, if it's ibuprofen or acetaminophen or whatever it is. Next, we have the ependymal cells, EPENDYMAL. There's two different kinds of ependymal cells, ciliated and non-ciliated. What they do is they mix and make something called CSF, which stands for cerebrospinal fluid. And the ciliated ones actually use the little cilia to move it along. So when we talk about the brain in Module 8, we'll look at how the cerebrospinal fluid surrounds the brain and cushions it and keeps it from being damaged, your brain from being damaged. If you get an injury and there's damage to the cerebrospinal fluid, that's called a concussion. Exactly. So the backup quarterback for the Dallas Cowboys got a bad concussion. So he's been out for several weeks now from a concussion. Yeah, so he was on the ground unconscious.
Oh, I
Face
thought
plant.
you were talking about the other one where
No, he broke that's the starting quarterback. So our second string quarterback is also out. So now we have a third string and a fourth string that are doing terribly.
Oh, you know, their time's charmed. No, it's not.
So maybe the backup guy will be in. They had a bye this week, maybe next week. Okay, so now that's 1, 2, 3, 4. So there's a fifth one I'm going to talk about. The fifth one, though, I'm going to talk about is in the PNS. So you may want to make a little note that those are in the CNS. And now we'll go to the one in the PNS, the neuroglia in the PNS. Are we all good to move to that one? Called Schwann cells. So we already know these guys. Okay, Schwann cells. That is the neuroglia for the PNS, for the peripheral nervous system. Okay.
For the what?
What is
The
function?
neuroglia, the fifth neuroglia, right? Schwann cell, PNS, only PNS. Okay? And what they're making is myelin sheath. Remember I told you that already in your picture? So here's the cell right
That's
here.
their function.
Their function is to coat this around, the myelin sheath around there. What the myelin sheath right there called myelination is it works to insulate the axon. It's insulating the axon.
So, like, to protect in a sense?
Yeah, okay, so think about a wire, So think of the axon like a wire, and a signal passes through it, right? Electrical signal passes through it. What do we cover our wires with?
Rubber.
Why?
Or electrical tape.
So we don't.
So they don't short out, so they don't get too hot. That's exactly what's happening on here. What the myelin is the mylin's the plastic around the wire. Okay? It's the plastic around the wire.
So protection of the.
Protection, yeah, it's also insulating. Insulating means that it's not letting that electricity get too hot, if you want to say it that way. But we're going to see it has so many cool things that it does. Okay, and that's what it looks like. On your picture of your neuron, it was those little packages all the way down the axon. That's your myelin sheath, being made by the Schwann cell, which is the neuroglia of the peripheral nervous system. All right, this right here, there are the Schwann cells right here. Okay, the Schwann cells. There are also something called satellite cells that are covering up the body a little bit right there. But what it's doing is, don't worry about those. We're just covering those to show where we have our insulation wrapped around there like our plastic. But we do have those little spots in the middle. Remember what that's called? A little gap in the middle called the node of Ranvier. Yeah, the node of Ranvier. Right here's the node, right here. That's where the sheath kind of ends. So the sheath is here. Here's our Schwann cell making the sheath. There's our axon. Look at like the wires there. And it turns out that little node right there is very useful. And it's going to be useful because it's going to help us with the speed of the signal going down. We're going to wrap it all up right here. We're looking at our neuroglia here. So on this side in the purple area right here, we're looking at things found in the central nervous system. We have our epidermal cells. They line the ventricles of the brain. The spinal cord. They assist in producing, circulating, and monitoring the cerebrospinal fluid. We have ciliated and non-ciliated right there. We have our microglia. They remove cell debris, wastes, and pathogen. Remember, diseases, right? Bacteria. Our astrocytes are maintaining the blood-brain barrier. They also provide structural support. They're kind of like a net that holds on to things. They regulate ion, nutrient, dissolved gases, and dissolved gas concentrations, absorb and recycle neurotransmitters. We'll get to that in a minute. Form scar tissue after injury in the brain. So they grow, and hopefully help support what brain tissue was actually lost. All right, then we have our oligodendrocytes. That is myelinating the CNS axons, and that is serving as insulation, and it says here framework right there. Now, over here on the left in the yellow, we have our neuroglia found in the peripheral nervous system. Mostly we're concerned about the Schwann cells right here. They're responsible for myelination and also for repair. We'll look at that in a minute. These satellite cells, which we don't care so much about over here, they help with regulation of gases, blood gases and things like that. Okay, so that's just a wrap up of that. All right, so we're moving now to nerves. Are you on the right in your book? Are we good? Nerves? Okay. So before you draw anything, let's talk about them. Nerves are bundles of axons and their sheaths. Okay, think about it. Look at your picture of a neuron. See that axon coming out there? All right. So and with a sheath around it, if I take those and bundle those all together, I've made a nerve. Okay, I've made a nerve now. Not the soma, not the body, but the axons with the myelin sheath. Yes? Okay. They extend out from the CNS depending on if they're from the brain ones or the PNS from the spinal cord, right, from the spinal cord. They're wrapped in shiny white epineurium made of collagen just inside the Schwann. I think it should say just outside the Schwann. So what I have here is I have the axon, which is covered by Schwann, right, which makes our myelin sheath, which is covered by endoneurium, which is covered by perineurium, which is covered by epineurium. All right, so what I think you're supposed to draw something around these sides. I think I have something else I want you to draw. Here's what I want you to know. Write these words down. Epineurium is the outside. You could draw a circle.
Where
Epineurium
You want us to draw
in that box.
In that box?
It is a box, right?
Yes.
Yeah. So it's
draw a what?
Well, so you're going to draw a circle that says epineurium. That's the big outside of the nerve. Right, draw your piece of sushi, okay? Remember, like our muscles, right? So we're going to have our big one like this, and that's going to be our epineurium. It's wrapped in that. And then we're going to have several on the inside, but let's just draw one like this. That's going to be my perineurium covering that. And then and then my actual cell, my Schwann cell, my axon are down in there. So you can see perineurium.
God, your mind way too small.
Endoneurium, perineurium, epineurium.
Oh my gosh.
I know.
Why are you laughing so?
So it's
She said, oh, it's really small and it's like that.
Epineurium's on the outside, then comes the perineurium,
That's funny.
then inside that is the endoneurium, then is our actual cell, our actual neuron. It's just like how we did the fascicles with the muscles, right? Or a piece of sushi. I have another picture here, but I don't think it's any easier to draw. Okay, here are our nerve classes. This is easy, right there. 3 nerve classes. We have sensory nerves. They are responsible for your senses. We have our motor neurons that are responsible for your motor. And then we have mixed neurons, which are responsible for both senses and movement. The vast majority of our nerves are mixed. But there are a few. For instance, your optic nerve coming from your eye is strictly sensory. The only thing it does is take light information and take it to your brain to be interpreted. That's it. So there's no movement involved in that one. It doesn't move anything. It's not part of your muscular system that moves your eyes around. It only takes light stuff. Motor, I don't know if I can think of one that's strictly motor. Maybe your book tells you that. But for instance, one of the things, I think in the book it tells a story about somebody who cut their hand and the nerve was damaged. And there was a loss of feeling and a loss of function because that was a mixed nerve. Okay, so I can't move it and I can't feel. And then if you get some work done and you line things up just right, then you can get some healing, sort of. We'll talk about healing in a minute. All right, so you got those classes written down? All right. Here's that sensory and motor right there. So it doesn't mean that all sensory are this, but they do look a little different than our regular old, what do we call that, multipolar? What was this called? What was this one called, bipolar? Or?
that was the unipolar.
Unipolar, with the body hanging off the side like that. All right, so talking about healing. Healing, there can be healing in axons of the peripheral nervous system, not the central nervous system at all. Brain damage is brain damage forever. But in the peripheral nervous system, if I can get these lined up, so let's say I have nerve. Remember, the nerve is a lot of these bundled together, right? A lot of those bundled together. So if I have nerve, if I cut my hand and I've cut the nerve right there, then what the surgeon can do, the surgeon can go take that nerve, which is much more visible than a single axon. It looks like a shiny shoelace in there. It's kind of white and shiny. Okay, and if you line them up kind of where they're sort of back together, we can actually have a little healing that takes place. As long as the soma's alive, okay, we can get the ends lined up, and there can actually be some growth, and it really isn't the axon that really fixes everything, it's actually those myelin sheath. The myelin sheath is kind of like a Band-Aid. The axon's never ever connected again, but because that Band-Aid goes around there, we can actually have conduction of a signal, because we're going to see here in a minute how that signal goes, and it conducts all the way down by jumping through the nodes of Ranvier. It's pretty cool stuff. Pretty cool stuff. No healing in the CNS. So that's what the blank says under healing, under nerve regeneration, only if they're lined up, and only in the PNS. Okay? No healing in the CNS at all. All you can have is some of those neuroglia that can sort of, just like the Schwann cell is making the myelin sheath, that's going to come alongside and help that nervous tissue of the axon. The axon's not going to regrow, but the neuroglia will. And that you can have some healing that way. Same thing in your brain, but it's only the neuroglia that are going to be doing that. The neurons themselves, gone.
So what do we put in the nerve regeneration?
Nerve regeneration only in the PNS, only if lined up, and that usually takes a surgeon.
And only if the soma is alive?
Only if the soma's alive. If the soma's dead, then we're done.
Only in PNS.
On PNS,
PNS.
peripheral nervous system. Only in the PNS, only if they're lined up. Typically surgically.
I don't sure how to spell live, that's sad.
And living soma, right.
And under healing, we just put
Under healing, it's pretty much the same thing there. Only in the neuroglia. All right, and at the bottom of page 42, it says experiment 7.1, neurons and neuroglia. That's microscope work. We're going to skip that. We probably won't even do it at our lab for 7. We'll come back in 8 to do that. All right, here's what, I don't know if you thought this might be easier to draw in your drawing thing. So where we have our epineurium, our perineurium, and then our ones inside there, right? So I didn't think this was any easier to draw.
I think the sushi.
Sushi's better.
yes.
Yeah. All right, we're going to 7b now. We're going to be talking about action potentials, exclamation point. What do I call action potentials?
Signals.
Signals. Signals. Right. All right, this is the hard part of the module. It's pretty hard. Yeah. Yeah. All right, so the action potential, here we go. There are 2 types of excitable cells. We know that. Neurons, not neuroglia, by the way. Only neurons and muscular tissue. Excitability, here's a word you're going to be filling in. Excitability is the ability to create an AP, a signal, an action potential, whatever you want to call it, in response to a stimulus. In other words, the ability to send a signal from the brain or to the brain. To or from the brain. Next word is not a word. Okay, there's an electrochemical phenomenon that occurs where ions cross the cell membrane, changing the electrical charge in millivolts. Yikes. And that's going to be looking at the potential difference. Here's the blank you're filling in. A measure of the charge difference across that cell membrane. So we're talking about electrical charges here. We're talking about electrical signal being sent. The electrical signal will turn into a chemical signal at the synapse. So the electrochemical phenomenon. Ions are going to cross the cell and change that potential difference of charges. And during this phase, is that what you're writing? What happens in this phase? Inside the cell is negatively charged. That should say charged, not charges. The inside is positively charged. That creates something called polarity, which gets reversed when electricity hits it. It's like throwing a switch. Boom. So remember, in 2 weeks we'll have a quiz over this, so I would highly recommend doing your study guide for this one.
Did I give you a study guide from last week?
I might actually have it. No, I think I gave it back to you.
Okay.
I think I gave back to you what I had. They were tests, I
Inside the
thought.
cells are negatively charges.
It said say charged.
Okay.
Inside the cell is negatively charged. There are negative charges inside the cell. And we're talking about a neuron here. Inside the cell, the neuron of a cell, the neuron cell. Outside the neuron cell, it's positively charged. There's polarity there. There's a attraction there. There's a, when I say attraction, it's like electrical whoosh. There's fixing to be some lightning in there. Okay?
So is the inside or the outside positive? 'Cause
The inside is negative. The outside is positive. Good point.
so the
Let's
outside
see.
is positive.
Let me make sure I got that right.
I already wrote outside. I crossed out inside, so that's right.
The inside is negative. The outside is positive.
Oh.
They both say inside, don't they?
Yeah. Oh.
It should say inside negative, outside positive. Sorry. Thank you for pointing that out.
Can I go pee?
Wait, don't leave.
Okay. This is the hard part. You gotta
Oh my gosh. We're going to get to it here in a second where I think you're going to be filling in a chart or something. Okay, so we're looking at polarity. We're looking at negative charges and positive charges across a cell membrane. Okay? Which brings us to rolling the
Action potential to stimulus and response.
I was about to say.
We're still writing. Okay. It brings us to the sodium potassium pump. And here's where you do this. Yikes. This will strike fear into the heart
if
of
you're actually
undergraduates.
saying this is hard, then that's kind of
Mm-hmm.
Yeah.
Mm-hmm. Yeah, this is not fun, y'all. This is not fun. Okay.
No.
You hate it when the teacher even is like,
I hate
it's
it when
not
the teacher
fun.
is serious.
Yeah, but my job is to try to distill it down to simplify it. That's what I'm going to try to do for y'all today, okay?
And then you
Down
could
to its
say,
finest
you know
place.
what, we learned it in class, the quiz, I think I did a good job, we want to.
No, we don't need to put this on the midterm.
This is almost to the same level as Krebs cycle, y'all.
Okay.
Here's Krebs cycle, here's
I
sodium
hate it.
potassium pump.
We barely even talked about that last year.
Oh, well, don't worry, in module 14,
Okay, well, let's
we'll
look
talk
at
about it a lot.
teacher
You're
without
kidding.
absolute
That's the most difficult
trash.
module in the whole year, is when we talk about Krebs. All right, so we have our sodium potassium pump. Now here's what I need you to understand. In the axon of the neuron, the axon, that long thing going on right there. Okay, we're talking about the cell inside. It's like a tube. There's inside the cell, there's outside the cell. Okay? So, at the resting potential, which means it's ready for a signal, a negative charge will fill the interior of the axon with large proteins, charged proteins, and the outside is positive, it's therefore polarized. Okay, so inside the cell, this is the inside of it, there are already these large proteins in there which are negatively charged. They stay there the whole time, they're gonna stay. Okay? So it's gonna be negatively charged. So the inside of the cell is negative. All right. The active transport of positive ions, sodium and potassium, the sodium potassium pump, serve to rearrange the ions, changing the polarity, allowing for the electricity to flow. All right? Now I have 2 videos that are going to introduce us to the sodium potassium pump. All right? So Danielle, I would say for the first one, wait, and if you can't wait for the restroom, then the second video, you can, yeah. The second video is terrible. I mean, terrible quality. The information's fabulous. Okay, but here's the newer one. Oh, okay.
Active transport is a type of cell transport that requires the input of energy in the form of ATP. The proteins that conduct this form of transport are often called pumps because they force molecules or ions to move from an area of low concentration to an area of high concentration. This is commonly referred to as against the concentration gradient. One of the more common examples of active transport is the sodium-potassium pump. The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell.
They're both plus.
Let's take a closer look at how this happens. The sodium-potassium pump has binding sites for 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split, releasing phosphate. This phosphate binds to a location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released. Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle. Notice that for every 3 sodium ions leaving the cell, 2 potassium ions enter.
You could say only 2.
Since both sodium and potassium ions have a positive charge, this unequal movement causes an electrical gradient to develop across the plasma membrane of the cell. A number of cellular processes, including the generation of nerve impulses, use this electrical gradient.
It seems to make like decent sense. Like it
Good.
doesn't seem overly.
Good.
Well, I mean the video made it seem easy, but
That's my goal. Okay. So it's kind of weird that they're both positive, all right? But so remember what's inside the cell? Some negatively charged proteins that are going to stay there. Okay, so the idea here is that I'm getting all the sodium out, okay, to make this negative now, and then we're going to end up flipping. And that's going to allow electricity to flow. Okay? All right, so this is the not so good one. It's mostly quality. The stuff it says is perfectly good. It's just.
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in, while other channels allow some flow of potassium ions back out of the cell. The sodium ions cannot easily get back in to replace the lost positive charges.
It takes a pump.
The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location
going
that
back
the
in.
membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative
See how
charge.
it turned positive?
This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside of the cell.
Okay, so you see how I was flipping that negative, positive, negative, positive, negative, positive? Okay. So interestingly, that first part we saw was actually the pump restoring the balance. I'm sorry, the second part was the pump restoring the balance. So when the sodium goes back in, that is the pump doing that actively with active transport.
So
I
Okay.
remember part of this from last year, or the year before.
Really?
General science or something. Was it general science?
Maybe. We did talk about the body, human body in general
I didn't
science. So.
do.
Okay, so now you have a box. You have a box that says, oh no, at the bottom of page 43, see where it says the sodium potassium pump? Okay, you're going to start filling in those blanks with the steps of the sodium potassium pump. This is going to give you step by step, okay? Yes?
I think it kind of confused me. So we're wanting the inside to be negative.
The inside's always negative. Always negative, 'cause protein's in there.
Uh-huh.
And we're wanting to turn it positive and then back negative again.
We're wanting
Boom,
to
boom,
do
boom,
that
boom,
so the
boom,
action
boom.
potential can go through.
That's how it goes through. It's exchanging that negative positive is changing back and forth, which is the electricity going through there. Remember, so the negative,
Positive inside for the action potential to go through.
yeah,
And then
so
it
we're
has to
gonna,
switch back to negative
yes.
to go back to normal.
well, right, to push it, we can push
Yeah,
it along.
push it.
We're pushing it along, okay?
Okay.
So flip,
So
flip,
when
flip,
it was
flip,
doing
flip.
that, that's like the action potential going through.
Yes, traveling down,
To send a signal.
yes.
Okay.
Okay, so the signal's being sent. So
Okay.
signals are coming through this wire right here by electrons traveling through it. In our wires, what's happening is the positive, negative are changing places, and that's the electricity going through it. Okay? All right, so let's go through the steps. Okay, so this is important. Number one, a stimulus is received from the brain. Either we touched something, we saw something, we tasted something, we felt something, a signal is received, and we have to respond to it now. So, a neurotransmitter is released. In muscles, we know it's called acetylcholine. We know that.
Wait, does it work from the brain or just?
Ultimately, yes, it's coming from the brain.
Wait. Oh wow.
Yeah, so a neurotransmitter is released. Okay. Sodium channels open up and sodium floods into the cell. So when I showed that to you, that first video I showed you, showed it going out, it was because it was reestablishing where sodium is supposed to be. So sodium channels open, sodium floods in, and we have a charge called negative 85 millivolts. Okay, we can actually measure it with a voltmeter, but it's millivolts.
So in the first video, only like at a time 3 can go in and 2 can come out.
Right, right. So that's just showing you that the ratio is, because since they're both positive, you think, well, isn't it going to be equal? No, we're going to continue to make that very negative. But we want it to be negative. So that was reestablishing the negativity of the inside of the cell.
Okay. So this process is like very quick.
Very fast.
Okay.
Very
'cause
fast.
This make it sounds like
I
it's
know, the
super
video
slow.
was
I know.
and then
Like,
So really,
yeah.
all these steps that are going on through here, why do there have to be so many steps? This is called the cascade of biochemical events. Yikes. Okay? It's just God showing off. He's just showing off.
Wait, what is Na? What is
Sodium.
Sodium.
Na is sodium. Yeah, for those of you who have not had chemistry, Na stands for sodium. Potassium, that's K, by the way. Neither of those actually have the letter that says the name, right? Potassium stands for K. K stands for potassium. So sodium channels open, the sodium floods in. It's positively charged. Well, potassium was already in there, and it's still in there. So guess what? We're having this kind of buildup of positive charges counteracting the negatively charged protein that's already in there. Okay?
Making both positive.
Number 5, depolarization. And this is where I put the word boom. So it goes from negative 85 millivolts to this positive charge of plus 30 millivolts. That's a big difference right there. So it's 85 plus 30 more. So that's a big change in the polarity. Do you see that? Where it used to be negative 85 between them, now it's positive 30 between them. Sodium channels now close. Potassium channels open and the potassium floods out. And it floods out so much potassium. Remember now the sodium's stuck inside. Sodium is stuck inside. Potassium's going out all the way till it repolarizes at, instead of negative 85, it goes all the way, it kind of overcompensates and goes to negative 90 millivolts, reestablishing that negative charge inside the cell. Lots of potassium leaving. Now the sodium is inside and the potassium is outside. We call that repolarized. But they're in the wrong place. They're in the wrong place. Where is sodium supposed to be? Outside. Potassium is supposed to be inside. Now they're in the wrong place. So guess what's going to happen now in step 9? Sodium potassium pump. It's going to replace them, put them back where they belong. It's going to put sodium and potassium back where they belong, so sodium in, potassium out, and it's going to restore that charge to negative 85 millivolts. And now it's ready to have another signal come through.
So if this is on the quiz, do you want us to have like the 30 MV up there and everything?
I need you to know what's happening. Okay, this is on page 197. I think that's the right page number. That might not be the right page number. That might be the old book. So don't write that down. Just know that this whole series of steps is in the book, okay? It is still called figure 7.8, I think. Okay, but these are the steps, and then we're going to go through a recap here. Another video.
Wait, so just 1 through 8 is the process of while the signal is being sent through.
Correct, that's
And
the
then
polarizing.
nine
That's
is changing it as it's finished.
back to
going through.
normal.
Okay.
Yes. So technically, the sodium potassium pump is only working right here, but we call the whole thing the sodium potassium pump. But technically, the pump using ATP because it's active transport is that step number 9. Did y'all have 9
It
bases?
was only 8.
Only 8. Sorry.
So it doesn't take ATP to do the process.
It does take ATP. It's all active transport. Getting the sodium out is also active transport.
Okay.
This requires lots of ATP.
Okay. So now can I go to the
If you want to miss the next video.
I drink coffee. It's like here's horrible. A super fast process, but
Yes.
it's like super complicated.
It's all happening very quickly, but
So
a lot of biochemical events.
the ATP just makes that sodium and that
Makes those
potassium.
open. Makes those doors open.
Okay.
Yeah. Are we still writing these steps?
So do you want us to put like the negative 8.5 B and everything?
Well, I'm gonna show you a graph here in a second that might be more helpful there. Okay? Let's watch a recap.
And sodium ions rush into the cell. This causes the cell to depolarize. It becomes positive on the inside and negative on the outside. Very quickly, the sodium channels close while potassium channels open, allowing potassium ions to rapidly diffuse out. This repolarizes the cell. It is back to positive on the outside and negative on the inside, ready to transmit another signal. Meanwhile, the sodium ions inside the cell have diffused to adjacent areas, causing a slight change in the polarity of the membrane ahead of the action potential. This change in polarity causes the sodium channels along this part of the membrane to open. Again, sodium ions rush in, and the action potential spreads to the adjacent part of the neuron.
Positive?
In this way, the action potential travels down the neuron like a wave. In the wake of the action potential, potassium leaves the cell to repolarize the neuron, and the sodium-potassium pump quickly reestablishes the resting potential distribution of sodium and potassium ions.
Notice I said it quickly restores it. Quickly restores it. Okay. Right, did that help you at all?
The sodium is supposed to be outside.
Correct. Is that right? Yeah, we're gonna get sodium out of there. Sodium's outside, potassium's inside.
So potassium starts inside, and then
Yes.
the sodium's outside, and then it switches.
So,
and
and
that
then
makes
sodium,
it
sodium,
positive.
sodium, some potassium out, and then we gotta switch them back. Yeah, all the sodiums and potassium stuck inside at the same time cause it to be positive. Then we gotta get the potassium. Why didn't God just take the sodium back out? Why does he send the potassium out and then he has to do because he's showing up. All right, this is what you're gonna draw in that little thing that says best shown with a graph at the top of page 44. You're gonna draw this. Let's see what's happening right here. Notice we have our cell right here. We have a message coming in through a dendrite. It's gonna flow down the axon right here. Okay, and when that happens is we get to a spot right here where our pump maintains sodium outside, potassium inside. That's what we just said, right? Sodium outside, potassium inside. All right, but a signal comes and we have all of a sudden it goes up with the sodium going in and in. It reaches the threshold. We're going to talk about threshold here in a second. And it goes up to where it's here negative. It says negative 70, but I think we had negative 85, yeah? Negative 85 comes up here all the way to positive. We have boom, and all of a sudden we have our change from negative to positive, a wild swing right there, and that's when the cell goes, okay, get the potassium out. So now sodium's still in, potassium's going out, 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 out. It's reestablishing now a negative charge inside the cell. It goes beyond the original, which was our 85, down here to 90. We call that hyperpolarization. And look what happens right here. What does you hear? See that word? Pump. It says here potassium out overshoot. Our pump is going to now reestablish where we have our sodium outside, our potassium inside. Maintaining. So this shows the whole thing of what you just wrote down in those steps.
Oh, is that where we draw?
You're drawing this, and it says best shown with a graph. You're going to draw this graph here. Big hump coming down below and then back up to the baseline right there.
So you want 85 instead of 70 though?
Yes, please. We will always defer to the book rather than Google. Yikes, we gotta get through this lecture, y'all. I got lots more to talk about. Okay, so, find that. I'm recording this whole thing. Here's our AP criteria. In order for the message to be sent, okay, you gotta have a couple of things. We're gonna be using the analogy of flushing a toilet. Okay? You just did that, presumably. Right? Okay, so here we go. It's all or nothing. All or nothing. You've gone to flush a toilet, sometimes it doesn't go, right? You're like, oh, that didn't work. I have to go flush it again. But once you flush it, can you unflush it? All or nothing. All or nothing. The threshold must be met or surpassed. It has to be a strong enough hit on the toilet thing to get the flush going. Strong enough. If you just touch it like that, that's not going to flush it.
Is this where we're...
Yeah. Threshold must be met.
We
The
ready to talk
string-
threshold
yeah.
stimulus?
Threshold stimulus must be met.
Yeah, let's see. Okay. The strength of the signal depends on the frequency of the signals, not the strength of the signal. That should remind you of muscles. Remember, muscles had to be the number of signals coming rather than strong muscle. Here we have, instead of a strong signal, we have a number of signals, a frequency of signals. The AP, or the signal, travels down the axon in a cascade wave. Here it is. Somebody asked about how fast. 300 feet per second. 300 feet, but that's fast.
Where do these go?
Hold
I
on.
got the threshold.
Voltage charge changes push the signal down the axon. We understand that. Voltage changes, flip, 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 right? Myelinated axons will increase the speed. All right, I think I have this somewhere, y'all. Okay, so just hold tight. I think I have it. This is just preparing you for that. So here it is. There's our toilet flush. Okay, there's our toilet flush. We have to get a strong enough flush to get it going. Once it's going, there ain't no stopping it. Threshold stimulus, here it is. A stimulus strong enough to trigger a signal. Strong, it's just a stimulus strong enough to trigger it. Strong enough flush. Sub-threshold stimulus not enough. I didn't push that toilet thing hard enough. Not strong enough to trigger the AT, the signal. Local potential is a small change in potential difference that will not travel down the neuron. So a local potential is too small, subthreshold. A refractory period, this is interesting. Refractory period is the time when the AP cannot travel down the axon because it's busy. So think about this, I just flushed the toilet, it's flushing, can I flush it again? No, it's in the middle of a flush. I go to try to hit that flush knob and it just blah, 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 blah. Right, nothing. It's floppy. That's what happens here too. So of course it's going 300, feet per second. So boom, boom, boom. But I have to wait. I have that few seconds of refractory period before, when I say few seconds, that a moment of time where another action potential cannot go. I'm limited there. All right, which leads us to speeding things up. We can speed things up here. Are we good?
No.
So Danielle, you're drawing. That looks good.
Oh.
Yeah. You just label it, when you get to your book or whatever. Label the parts, but yeah, that big old curve on there is good.
Yeah, I forgot that we were supposed to do that. I was just looking at it.
Yeah, so
I
it's
just have the-
totally in your book.
I just have the loop
And then you can label it as far as 85 plus 30.
That everything that you're showing is going to be in the book. So
It is all. It is all in the book. Okay. So something called saltatory conduction. So it turns out the myelinated axons, which is one of your words, right? Myelinated axon, that's an axon with myelin sheath on it. It turns out they can jump between the nodes of Ranvier. It can actually go boing, boing, like leapfrog. Instead of traveling all the way down that axon, it can actually leapfrog through those nodes. That's known as saltatory conduction. Non-myelinated axons carry the signal in continuous conduction. So there are some neurons out there that don't have any myelination on them. That's true. That happens. In fact, it might even be the majority, but okay.
So what do you want for myelinated axon?
Myelinated axon means it has myelination. We can cause that. We can have. I want you to write that word saltatory conduction next to it.
So how would
Saltatory
you-
conduction equals jump.
I just said increase the speed.
Increases the
That's what you
speed.
said earlier.
I just need you to know the word saltatory.
Yeah.
All right, so we can, for instance, if you have a sudden sharp pain, you just hurt yourself, that's called the saltatory, and later dull pain. You've had that where it's an injury where it hurts immediately, and then later on you kind of, that dull kind of a thing. That is
Sore.
the continuous, yeah, it's kind of sore, but like I'm throbbing kind
All
of
we
a thing.
need to write is solitary conduction.
Saltatory, it's jumping, makes it quick. So I want to tell you a little bit about babies. I was telling my sister just had my sister's daughter just had a baby, so my niece just had a baby, a little baby girl. And her name is Eleanor. And so y'all have been around babies, yeah? Okay.
All the time.
So what happens, you got this little baby, you know, they're all kind of bendy and all, because we know about their bones and everything like that. Well, what happens is none of their nerves are myelinated when they're born, none. That's why they're doing this. All their electricity is just, boop, 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 it's not going in the right place. They're just goofing like this, right? And so what happens is the myelination begins in the brain. So we have those oligodendrocytes, and now we have from the PNS coming and myelinating those ones coming out. So one of the very first myelinated nerves to be are the nerves that's going to feed the eyes. So one of the first things the baby can do is focus on mama's face. And they notice mama's face, right? And they recognize it. Then it comes down, what's one of the first things to do? They smile, right? They smile. And so that's because those nerves have been myelinated. They have control over their smile. Prior to that, you may have seen a smile, but it was some sort of, this kind of stuff. But now they actually smile at you. Okay, so then they get the myelination of their nerves moving down their neck. Guess what? They can hold their head up. All right, then it goes down to their arms. They can look and they see their fingers. They go, oh, and they start grabbing stuff. They see that their fingers are theirs. Okay, then it moves down and they can sit up. And it moves down to their legs. They can crawl. Eventually they can stand up. Eventually they can walk, right? That's because that myelination is working its way down the body. One of the very last set of nerves to be myelinated are the nerves that feed the bladder. That's why potty training takes longer. The nerves are just not ready yet. So really at least 2 years. Some people try to potty train their babies at 9 months, whatever. You gotta wait for those nerves to be
My
myelinated
aunt
right
tried
there.
at like 10 months and it completely backfired on her. My mom's like, that's because we just need to let the child wait until they're ready.
Till their nerves are ready.
Yeah.
Exactly, till
I
their
never
nerves are
thought
ready.
of that process being because of that. That makes
Yeah.
so much sense.
Isn't that cool? Yeah.
Of why like moving
And you can
is,
watch that progression of the myelination right there. Okay, so here's our cell. Here's our myelination right here. Let's see how it's jumping. Boom, boom, boom. Look at that little, we know that. We just drew that, right? We're going to see that boom up to that plus 30. Jump, boom, to the plus 30 that jumps past like this. Here's our node. So this, not only is it insulating the cell and doing all that cool stuff and protecting and everything, it's actually increasing the ability for that signal to travel down. Okay, so this next picture is going to show you, here's unmyelinated. Watch how fast that blue thing moves. It's done on the right, done. It's only not even halfway done on the non-myelinated one.
That's crazy.
Okay, so that's how much faster it is. And what kind of conduction is that called? Saltatory.
What kind of nerves don't have?
Some of the neurons in the PNS do not have myelination on them. So when I show the myelination, that's the representative of it, but there are plenty that don't. And so they'll just take longer. You know, I'm not sure why it's designed that way. I just know that in particular nerves, like when we have mixed nerves, we have some of the neurons going this way and some of them going this way, right? Because some of them are for sending a message, some are for receiving a message. That would be our sensory ones. And so they're going both ways, and some of those within there are wrapped up in that nerve, but they're not myelinated themselves.
That's the bladder one that you're talking about.
It just comes later. No, it's myelinated. It just comes late. It happens later. Okay, a signal begins at the axon hillock. That's right at the very beginning of the axon on your neuron right there. Travels down the axon and reaches the synapse. It's going to translate this information from one neuron to another neuron, or in the case of muscles, from one neuron to a muscle. And it's going to do that by crossing over an axon. We know about a synapse. We know about synapses. We did this in the last module, right? Or the module before last? Hold on. We have a presynaptic neuron and a postsynaptic neuron. There's a plethora of information assaulting our neurons every moment, so most neurons are inhibitory. They stop messages from being sent. This is the whole, I put on an itchy sweater, it's really, really itchy, but after I wait a little bit, it's not itching me anymore. because your brain says, I'm not going to feel that anymore. It's like the smell going nose blind, right? You stop smelling it because your brain says, why aren't they getting rid of that trash in the house? No. So your brain starts inhibiting that ability to smell that, or that ability to feel that itchiness on you. Association neurons, neuron to neuron, are tightly regulated by inhibitory synapses. Not like muscles, they're tightly regulated by inhibiting signals to go. So we have something called the excitatory chemical synapse, also known as the EPSP for excitatory postsynaptic potentials. All right, cascade of biochemical events, yay. A signal reaches the synapse, we have calcium coming in. We remember calcium from the muscles. It rushes in at the synapse. Okay, we're fixing to go from synapse to another, axon to another axon, or neuron to another neuron. Calcium channels open at the presynaptic terminal at the beginning. Synaptic vesicles release neurotransmitters in the gap, oftentimes acetylcholine will be for muscles. A neurotransmitter travels to the postsynaptic neuron and binds to a receptor that's already there. The receptor opens sodium channels, which depolarizes the neuron. Sodium channels, guess what's going to happen now? Sodium will go in and we'll begin our sodium-potassium pump process. Okay? But so how did that process begin? It began with in our thing we just said signal is sent. This is the signal being sent. And it's being sent through an axon. Through a synapse. Okay? Axon, synapse, synapse.
I'm supposed to read this.
Well, I don't know.
It says chemical synapse, but it's like a big box. Like a huge box.
Chemical synapse. I think you're going to draw something.
Okay.
Great.
Okay?
I'm sort of writing in it.
All right. So you want to draw that?
Oh.
Let me see if something else is better. That one?
I think the other one's better.
Yeah.
Maybe.
So notice the axon is touching a dendrite of another neuron. Okay, so here's the end of my axon, the bouton, right? And it's going to, remember it's not touching, there's a gap in there called the synaptic cleft or the synaptic gap. And it's going to go from an electrical signal to a chemical signal, which is now accepted on the other side if there's a receptor for it, which is going to, so what's going to happen is calcium's going to rush in order for these to come out. These will come in order to open up sodium doors. Sodium doors get opened up, they rush in, we have sodium-potassium pump situation happening. So this synapse should be familiar to you because we talked about it when a neuron touches a muscle. Okay, so I have to move on, y'all. I like this picture because it shows the entire axon coming down. Here's the body, here's the axon. I have no dendrites on there. I have no myelin sheath on there just because we're going to just ignore those for now. We're going to see how they're traveling down. Here is that chemical coming down there, right? So that is my neurotransmitter. It's going to come down. It's going to be dropped off over here, and the other are going to accept it right there. Let's watch. I'm going to skip that one, and we're going to do this one. This one's the newer one. It's better.
The nervous system is composed of billions of communicating cells called neurons. Neurons conduct electrical signals or action potentials along the cell's membrane, along the axon to the terminus, where synapses are formed. The synapse consists of the membrane of the presynaptic cell, a synaptic cleft, and the postsynaptic cell membrane. Since an action potential can only propagate along the neuron's membrane, a chemical known as a neurotransmitter must bridge the gap between the adjacent cells.
We knew that.
The presynaptic cell releases the neurotransmitters into the synaptic cleft, where they will move to bind the receptor proteins in the postsynaptic cell membrane. When the action potential arrives at the synapse, it opens voltage-gated calcium channels, resulting in an inward flow of calcium. The calcium binds to the vesicular membrane. This binding activates the vesicles to fuse with the presynaptic membrane. As fusion occurs, neurotransmitter molecules are released into the synaptic cleft. Once in the cleft, the neurotransmitter molecules diffuse throughout the interstitial fluid in the cleft. Some of the neurotransmitters will cross the synaptic cleft and bind with receptors in the postsynaptic membrane. This binding produces an effect in the postsynaptic cell by opening the protein channels and allowing the flow of ions into and out of the cell. If the effect of the neurotransmitter excites the postsynaptic membrane, an excitatory postsynaptic membrane potential, also known as EPSP, is generated. This localized depolarizing signal generally causes long-distance signals, action potentials, in the dendrite of the postsynaptic cell. If the effect of the neurotransmitter inhibits the postsynaptic membrane, an inhibitory postsynaptic membrane potential, also known as an IPSP, is generated. This localized hyperpolarizing signal generally prevents the formation of long-distance signals in the postsynaptic cell. Neurotransmitters influence the postsynaptic membrane electrical state for only a short time and are removed from the synaptic cleft in one of three ways: diffusion away from the receptors, enzymatically degraded, or recycled by the presynaptic cell. Some of the neurotransmitter molecules diffuse out of reach of the neurotransmitter receptors. Some neurotransmitter molecules are removed by enzymatic degradation. Some neurotransmitter molecules are transported back into the presynaptic cell by neurotransmitter transporter proteins, where they are recycled.
Okay, I'm gonna skip ahead because we are out of time. I'm just gonna, this is not a note for you to take right here, but talk about those inhibitory ones, remember, so that things are not being felt or whatever. We're gonna see how it's gonna cause this to be changed this way, all right? Successful transmitters, in order for an AP to be created, there have to be several APs, several signals releasing neurotransmitters, increasing frequency, increasing neurotransmitter, called temporal summation. And we're just talking about and spatial summation. I'm recording this. I may send it to you if you want me to. Spatial and temporal summation. This is in your book?
Yes.
Okay, these are what you're supposed to write down, the very last thing on your book right there. Our neuron arrangements look like this. This is what you're supposed to write. If you want to take a picture of that. It's right here at the bottom that says
I'll take
neuron arrangements. And it shows you we have divergent, where one message gets spread to the different parts of the body. Convergent, where many inputs lead to one limited output. And oscillating, the messages regenerate itself, and they look like this. So diverging, we have things that like a reflex action. Converging, we have tightly controlled output. Oscillating, long-lasting, for instance, our awake-asleep cycles.
Go back and look.
Yep, right there. All right, and that's it. And you'll have 2 weeks before you have to have a quiz on this.
Thank God.
All right, so have a lovely Thanksgiving.